Dr. P again for our second lesson now on enzymes. And when this second hour, what I want us to do is, rather than necessarily recapping, we've already kind of done that, let's really start to apply that knowledge into a much more kind of challenging scenario. And what you're going to find in the GCSE is that often these kind of enzyme experiment questions are very common, and they can be quite difficult for students, I find, over the years. So having a good kind of overall knowledge of kind of how science works, experimental procedures, as well as a little bit more specific detail on some types of enzyme control reactions and how they can be measured is going to be really useful to you guys. So it's going to be a trickier lesson this second half, but I think you'll get a lot out of it, and hopefully this will really kind of up our grades overall. But before we kind of really get into the nitty-gritty of linking experiments with enzymes, we need to make sure that we are all on the same page when it comes to some of the really basics of science and science experiments, stuff you did in year 7, but I find over the years that students, even though they get taught this again every single year, forget it. And I think it's very, very important that you get your head around that, and that is scientific variables. Now, there are 3 main types of scientific variables that we need to know about. The first is independent variables, then it's dependent variables, and then it's control variables. Okay? So an independent variable, the way I like to think about it and memorize it and make it easy is it's the thing you're investigating. It's the thing you're changing in the experiment. If I change this, what effect will it have? Okay? My dependent variable, of course, depends on the independent, and it's a thing that we are measuring the experiment. So if I change this, my independent variable, how do I know it's had an effect? I need something to measure, and that's what your dependent variable is, the thing you're measuring, so you can gauge the outcome of the changes that you're making. But of course, if you're changing something in an experiment and you get some results, if you don't control your experiment, make it a fair test, then there's a possibility that other factors, besides the one that you're actually changing, could affect your actual results. And you could be making conclusions that aren't actually valid. So in order to make sure that we can make some valid conclusions, we need to consider our control variables. Now these are things, multiple things, that we might need to control in our experiment to make it a fair test. Okay? There's usually going to be more than one. So you might want to have a look at either the method that's been presented to you in the exam question, or if you're writing your own, what would you need to control throughout of it to make it fair? When you're writing the method, you don't have to state these are my control variables. Just simply, state within the method, like I'm going to use the same concentration of enzyme throughout, or the same concentration of substrate throughout, or I'm going to control the temperature using a water bath, whatever it may be. Have it within the method to make it a much more high quality kind of grade 9 through to 7 kind of answer, rather than having to kind of shoehorn them in separately. So what I want to do now is actually sort of talk about how do those variables relate to those enzyme experiments. So that's what we're going to move on to next. Okay then, so let's have a look then at how we can think about our independent, dependent and control variables in the context of enzyme controlled reactions and the experiments we would use. So your independent variable, the thing you're investigating, the thing you're changing, generally speaking, you can factor this down into 5 kind of things that you could look at. You could either look at the effect of temperature or pH, because we saw in the previous lesson how they can affect the action of enzymes. So you could easily use either of those as an independent variable, or other things that you might not have considered yet, like the concentration of the substrate. So if I have a higher concentration, then in theory I should have more collisions between my enzyme and substrate, and the reaction should go faster. Likewise, the concentration of the enzymes could be another factor you can investigate. So effectively you have a higher enzyme concentration, there's more enzymes there, therefore more likely to get collisions and more enzyme substrate complexes formed. And then the 5th factor you could investigate could be looking at a substance that affects the action of the enzyme. Maybe there's something like how bile aids in the digestion of lipids and helps with the action of lipase, which you should have looked at in the plenary in the last lesson. Your dependent variable is obviously what you're measuring, and because an enzyme enzyme controlled reaction is a chemical reaction, your knowledge of chemistry and chemical reactions and how you measure those is going to be invaluable here as well. The same rules apply. So generally speaking, in a reaction, reactants form products. Simple as that. So either we can measure the disappearance of those products or the disappearance of those reactants, and the formation of those products. There are options. Many ways of doing that. You could use like colour changes and use those sort of food test chemicals like Benedict's, iodine, Biurea, whatever you like. We could, depending on the nature of the enzyme action, we could measure things like pH, how cloudy the solution is. Lots of things you could do there. But generally speaking, they're going to boil down to, are they measuring the reactant disappearing or the products being formed? That's simple. When it comes to your control variables, you want to link it back to independent. So you've hopefully identified what your independent variable is in a particular exam question. And then once you've identified that, the other factors I mentioned, you'll need to control. So for example, if your experiment involves changing the temperature and seeing how that affects the rate of reaction, well then you need to control the pH. You could use a buffer solution, which is basically a chemical that you add that maintains a particular pH. So that would control it, that will stop the pH from changing. Or you might have to control the concentration of substrate, because it wouldn't be very fair. In one experiment, you've had a low concentration of substrate, and then the next one you up the concentration. It's going to affect your results, isn't it? And if I'm investigating something like temperature, I don't want that as an extra variable confusing my results. I want to remove that as best I can. Likewise, control your enzyme concentration. That's how you do it. That's simple, right? So factor those in, remember that, and that will hopefully, a lot of those skills there will be kind of universal for both biology, chemistry and physics. So jobs are good on that one. Okay, so we're going to move on now from variables, and I might give you a few examples of some maybe things you might not have come across yet, particularly involving the action of lipase enzyme, and also introducing you to a bit of equipment called a colorimeter. Okay, so I want to talk to you about this bit of equipment here called a colorimeter. Now, first of all, don't panic. It's not something you have to know a lot about at GCSE or even very much at all. I have seen it come up a couple of times in GCSE exam questions, so I thought it's worth, with our kind of stretch and challenge goal in mind, to talk about it. Basically, it's a piece of equipment that measures the absorbance of light. So you shine light through a sample, and you've got a little detector at the back that can detect how much light is passing through the solution and how much is being absorbed. So the cloudier the solution is, the less light will actually pass through, and more will be absorbed, so we'll detect less light on the other side. The clearer the solution is, the more light should pass through, the less that's absorbed, and therefore the device, the colorimeter, can detect a higher concentration of light making its way through, higher light intensity, So how does this relate to enzyme control reactions? Well, let's consider this. Hopefully you've done this in chemistry already. You may be familiar with sodium thiosulfate, and when you add acid to it, it goes cloudy. And the way you do that experiment is to put a little paper cross underneath your conical flask, and you time how long it takes for that cross to no longer be visible. Well, what we're doing here is something very similar, but actually using, rather than a subjective measure like how long does it take, I can't see the cross, I can still see it, causes all kind of problems. By using the colorimeter, it's a much more precise, much more accurate way of doing things. So if I had an enzyme control reaction where, say, the substrate, maybe like protein or something, is in our sample, it might be creating a very kind of cloudy, kind of dark sample. As a result, we would have high levels of absorbance of light. Okay, but as we added the enzyme, like the protease enzyme, it would break down the protein into amino acids, which might not create such a cloudy solution. So the solution might become clearer and clearer, allowing kind of less and less absorbance of light. So our values would change. So that's one, kind of application of it. Of course, it could be the other way around. Our product could make things cloudy. Depends on the scenario. But the one thing I will stress to you again at GCSE, don't panic, don't worry too much about it. It does give you the information. at the start of the question about colorimeters and how they kind of work. So it kind of sets you up. Of course, you will have to apply your knowledge and think outside the box for that kind of level 9 kind of question. But I thought I'd give you a bit of a heads up, get you thinking about it in the right way. And there's a little exam question for you guys to do in there that will test some of that knowledge of the colorimeter. So now I want to look at how we can measure lipase in a slightly different way. Okay, this is a common question on lipase, so we'll cut to that in a sec. Okay, so lipase is an enzyme that breaks down lipids. Okay, now hopefully you remember that lipids get broken down into fatty acids and glycerol. So I'll put that on the board here so we can help with our little discussion that we're going to do. So lipids get broken down into both fatty acids and glycerol, and they're very particular of that in the exam. You know, very rarely will allow you to say one, you have to mention both, otherwise you might not get the mark. So one way that we can measure the action of lipase reacting, say, with something like milk, breaking down the fats, the lipids in milk, is by measuring the change in pH. Now, looking at this and looking at the products, hopefully you can spot what I can see, how I could use pH in this reaction. And if you're struggling a little bit, look at the first bit, fatty acids. Okay, so over here, I haven't got any acids. In fact, milk will be slightly alkali. But as the lipase begins to break down the lipids in the solution, more and more fatty acids are produced, which will make the solution more acidic. And the pH should drop, should decrease and become increasingly more acidic. This actually happens if you leave your milk out too long. The bacteria present in the milk will start to break down the lipids, and that will actually give you part of that sour taste, is the acidity increasing in the milk. So, you know, a real world application there. So using something like a pH probe, I could time how long it took for the pH to drop and eventually level off. Now I've got a question in the lesson, in the PowerPoint for you guys to consider as to why the rate of reaction would level off. What you'd see is something like this. So we'd have time along the bottom, our pH here, and the pH would start off high. Let's say I'm adding in the enzyme now, and then what we would see is that pH level would then slowly start to decrease, or in this actually case quite rapidly, and then actually eventually level off. My question for you guys is a sort of stretch and challenge point of view to think about. Why would that graph level off? I'm not going to give you the answer here, it's all in the PowerPoint for you, so you can test out your knowledge, but think about why that might eventually level off. Why would the pH no longer continue to decrease? Okay, so the last bit I want to talk to you about is possibly the hardest, putting together everything we've done so far into writing your own methods for an enzyme controlled reaction. So we're going to cut to that in a sec. So when you're planning and writing a method, possibly if it's like a 6-mark question, here's some advice. You know, of course, do your busy bee work so you can identify what you need to do and help you plan your answer for your 6-mark question. But remember that a method is, generally speaking, a numbered or a bulleted set of instructions. Do this, do that, do this. Pretty simple. You know, when you're baking a cake, you're following that recipe, aren't you, to the letter? Doing everything precisely as you go. Now, if the recipe wasn't very good, and it might be lacking in key information like how many eggs do I use, what's the massive sugar I need to use, if it doesn't actually say to add sugar or add an egg or add eggs, you might end up with a really rubbish cake. It has to be done very precisely. The ingredients have to be in the right, amounts. And likewise with a method, your step-by-step instructions should be as clear and as precise as possible. So in theory, anybody should be able to carry out your experiment. You should be able to give your method to a Year 7 student who doesn't know anything about enzymes yet, and they should be able to follow your series of steps and actually get valid results. Now, valid results is where your experimental data actually answers the original question that you're trying to test. So, for example, does temperature affect the rate of protease breaking down proteins? So, if your experiment is done correctly and you've done a range of temperatures and you've measured using some sort of technique, maybe like bioarray or a colorimeter, whatever it may be, to measure the end point of the reaction, and you're going to range of temperatures, then your data should be valid. It should help you see, how does temperature affect my rate of reaction? You should be able to find specifically which temperatures the enzyme work best. Now, if you've only picked 3 temperatures, then the validity of any kind of conclusion, as in what's the optimum temperature, will be a bit weaker. You know, for example, if I investigated 10, 20, 30 degrees C, and you might get some result, a graph that looks like that. So enzyme activity up here, temperature along the bottom. A conclusion might be that 30 degrees C is the best temperature for this enzyme, it's the optimum. But that wouldn't necessarily be valid, because is it possible that actually the rate could carry on increasing? So actually 40 degrees C might be better, or even something in between. So the more temperatures you do, and the higher the resolution you've got, the more precisely, the more valid your conclusions will become. All right. So when you're writing a method and you're doing your busy bee and so forth, I would think about what your variables are. What is the independent variable in this experiment? What's the dependent? What are the controls? You don't necessarily have to go, this is my independent variable, that's my dependent, etc. I don't think that makes a very good conclusion. But you might want to bullet them down, jot them down in the margin somewhere. And that's going to help you frame and think about how you're structuring your method. And like I said before, with your control variables, make sure you're using the same concentration of enzyme, the same concentration of substrate, temperature, pH, depending on which one you're doing as your independent, sort out the other ones and mention that in the actual question. You don't have to give specific quantities or anything like that. No one's expecting you to do that. But even just saying, I will use the same concentration of enzyme at each temperature, or whatever it may be, will be absolutely sufficient, and it will show to the examiner that you understand how to control an experiment. You've written down some step-by-step instructions that anybody can follow, and in theory your method should produce valid results. So the final part of the lesson is basically for you to put all this together, all this information, and first of all, plan by doing a busy bee on the 6-mark question, and then writing your final answer. I've included the mark scheme at the end of the PowerPoint so you can check your answers once you've completed it, and hopefully you've done a good job. So this is Dr. P signing off for lesson 2. I'll see you later in the week. Cheers.